Hello dear friends, I hope you are all fine and watching this video in perfect health. So as we can see, this is a question from 2016 biology paper 2, and the question was on enzymes. Okay, so the first thing in biology is you study the diagram that has been given to you. So looking at this diagram, the question is saying figure 2.1 shows the effect of pH on the rate of enzyme catalyzed reactions K and M. So we've got enzyme catalyzed reactions, reaction K and reaction M. Okay, and both these reactions are reacting in different mediums in terms of the pH value. Okay, so as we can see, there is pH concentration, then there is rate of reaction. So as we can see, there is according to the pH scale, from 1 to 6, that is an acidic environment. So this environment is acidic. Okay, then from 8 to 14, this is an alkaline environment. So alkaline environment. So these two reactions are taking place in two different environments in terms of pH value. So pH means how acidic or alkaline an environment or solution is. We call that as the pH. So pH is how alkaline or acidic an environment or solution is. Okay, so that is what we have for the diagram. Okay, so now that we have seen and observed what we have here, we can therefore start answering our questions. So the first question is saying, A, what term is given to pH at J and L? So what do we call the pH at J and L? So as we can see, this is where the enzyme activity is working at its best, because these two reactions are being affected by enzymes. So at J and L, the enzymes are working at their best. And so in terms of pH, a point where the enzyme is working at its best, we call that as the optimum pH. So we are going to call that as the optimum pH. So optimum pH. Yes, so we call that as the optimum pH. If it was temperature instead of pH concentration, if it was temperature in the horizontal part, would have said the optimum temperature instead of pH. Now since it is the pH scale in the horizontal part, we are therefore calling this as optimum pH. So the optimum pH or the optimum temperature, this is a region where an enzyme works at its best. Okay, so enzyme activity is at its best at optimum. In this case, we have J and L. Okay, so let us now proceed to B. Okay, so B is saying, explain why the rate of reactions K and M goes down after point J and L. Actually, even before point J, the reaction is not that much at its best. Even here, before point L, the reaction is not at its best. So now, what does this mean? It means, for example, on the acidic part, this enzyme works best at 2.4. After that, it reduces in terms of activity. Same applies to a reaction M. This enzyme works best at 11. After 11, its activity is reduced. because it does not work well in too much alkali. Okay, so how are we going to answer this? So we're going to say because enzyme activity was reduced. Enzyme activity was reduced. So the enzyme becomes enzyme activity is reduced after 2.4, 2.3, even here after 11 the enzyme activity is reduced. So that is all you needed to get the 2 marks, because enzyme activity was reduced. So let us now proceed to question C, and C is saying name the regions of the alimentary canal where reactions K and M take place. So what regions can you find a reaction K and M? And from that diagram we can see that reaction K is in an acidic environment, where reaction M is in an alkaline environment. Now the environment is determined, the pH value of the environment is determined by the type of juice that is produced. Okay, so for example, in the stomach, gastric juice is produced, and gastric juice has got hydrochloric acid. So meaning the stomach is acidic. So we're going to say stomach. Stomach is acidic. Then in the mouth is alkaline in nature. Same applies to the duodenum. These regions are alkaline. So we're going to say the mouth and the duodenum. That is the first part of the small duodenum. Okay, that is the first part of the small intestine. That is where you can find the reaction M, which is alkaline, and the stomach is acidic. That is where you can find a reaction K. Okay, so that is straightforward and simple. D is saying suggest one enzyme which can catalyze reaction M. So the enzymes that can catalyze reaction M, meaning a reaction taking place in the mouth and the duodenum, and these environments are alkaline. So what enzymes? So in the mouth you find salivary amylase, in the duodenum you find the pancreatic amylase. So amylase is one of the enzymes that catalyze reaction M. So we're going to say amylase. Some will pronounce it as amylase. So apart from amylase, you also have trypsin in the duodenum. So trypsin, and then you also have lipase in the same duodenum. You have lipase. So meaning the enzymes that catalyze lipids. Trypsin is a protein enzyme. Amylase or amylase is a carbohydrate enzyme, and we've got 2 types of amylases. So we've got salivary amylase and pancreatic amylase. So these enzymes, they work well in an alkaline environment. So when they say suggest one enzyme which can catalyze a reaction which takes place in an alkaline environment, such as reaction M, we are going to say amylase, which is salivary, trypsin, and lipase. So amylase can be salivary or pancreatic. Okay, then D2 is saying state one food nutrient which can be catalyzed in reaction K. So reaction K usually takes place in an acidic environment, and the only region during in the digestive tract which is acidic is the stomach. So a food nutrient which is digested or catalyzed in the stomach is the protein. So chemical digestion of proteins begin in the stomach. So we're going to say protein. Protein is the food nutrient they want. So we're going to say protein. Okay, so protein, that is the major one, because it takes place in the stomach. Chemical digestion of proteins begin in the stomach. Then E, state two factors other than pH which affect enzyme activity. So we are going to say temperature, okay. We also have concentration of enzyme, so concentration of enzyme, also concentration of substrate. You can also say inhibitors, as well as the cofactors. So enzyme inhibitors or enzyme cofactors. So that is how you are supposed to answer this question. Okay, so let me even put the other one. So you can say enzyme inhibitors. You can also put the enzyme cofactors. So enzyme cofactors. Then on concentration, you can also put substrate concentration. So apart from concentration of enzymes, you can also say substrate concentration. Okay, so that is how this question was supposed to be answered. That is very simple and straightforward. Biology is one of the simplest subjects, provided you study it almost on a daily basis, because the nature of the syllabus is very, very, very big, complex and complicated. So what you need is to study each and every day to keep yourself reminded. Okay, so if you enjoy the video, or if you have learned one or two things, I advise you subscribe to our YouTube channel. You also follow our Facebook page, which is the Northern Gates Tutorials. Okay, so thank you very much for your time and stay blessed.